Hello, and welcome to the Sleepy Curiosity channel. As you find a comfortable position, let your body begin to release the weight of the day. If this gentle journey is already starting to slow your thoughts, consider subscribing now, so these stories can keep finding you night after night. Now, let the relaxation settle into your breath. Inside every cell of your body there's a text with 3 billion letters. And a single wrong letter in that text can completely change a human life. This isn't a metaphor. It happens every day, in every birth, in every cell division, in every generation that passes. And the more we look at it, the more remarkable it becomes. There's a question that puts experts in very different positions from one another. What exactly is a genetic disease? Most people picture something inherited from their parents, something that comes programmed before birth. But that definition leaves out situations that are also, technically, genetic. And it includes others that don't always show up the way you'd expect. A genetic disease is any condition caused by a change in a person's genetic material. That change can be present from the moment of conception. It can arise spontaneously in some cell of the body over the course of a lifetime. Or it can be inherited from one or both parents. What defines a condition as genetic isn't necessarily inheritance. It's the origin in the DNA. The detail that surprises a lot of people is that not every genetic disease is hereditary. And not every hereditary disease is genetic in the way most people imagine. A cancer that arises from mutations accumulated over decades is genetic, yes. But it wasn't inherited. Meanwhile, a predisposition to diabetes can be inherited without ever becoming a disease, depending on the person's lifestyle over the years. This starts to get more interesting when we realize the scale of what we're talking about. The human body has approximately 20,000 functional genes, distributed across the 23 pairs of chromosomes found in nearly every cell. That number, curiously, is smaller than that of some plants and even certain worms. What sets complex organisms apart isn't the number of genes, but how they're organized, regulated, and interact with one another. Each gene works like an instruction for producing a specific protein. When that instruction contains an error, whether it's a swapped letter, a missing sequence, a repetition where there shouldn't be one, the resulting protein may not function correctly. Or it may not be produced at all. Depending on which protein is involved and what role it plays in the body, the consequences range from completely imperceptible to profoundly serious. And there's a detail that changes everything. The same gene can cause completely different effects in different people, depending on what other genes are present in the body, the environment the person lives in, and even the moment in life when the change occurs. There's no simple, direct relationship between gene and destiny. Not every cell in the body has the same DNA active at the same time. Even though they all share the same genome, each type of cell uses only a specific fraction of those instructions. Neurons, liver cells, skin cells, each one reads a different part of the same book. This means a mutation in one gene can cause problems only in a specific tissue, even when it's present in every cell of the body. This selectivity explains why certain genetic diseases affect a single organ, even when the mutation is present throughout the body. Huntington's disease, for example, is caused by a mutation present in every cell of the body. But it primarily destroys neurons in a specific region of the brain. The exact reason is still under study, but it involves the fact that that gene is particularly active in that type of cell. What almost no one realizes is that this has direct implications for developing more precise treatments. Understanding which genes are active in which tissues is one of the most important frontiers in genetics today. And every answer found opens up new, even deeper questions about how life organizes itself. There's a distinction that rarely comes up in everyday conversation, but that's central to understanding genetic diseases. It's the difference between genotype and phenotype. The genotype is the set of genetic instructions a person carries. The phenotype is what actually manifests, the appearance, the functioning, the visible symptoms. And the distance between these two things can be enormous. Two people can have the exact same mutation and present completely different clinical pictures. One may develop severe symptoms in childhood. The other may reach adulthood with no perceptible manifestation at all. This phenomenon is called variable expressivity, and it stems from the interaction between the altered gene, other genes in the body, and environmental factors over time. There's also the concept of penetrance, which is the probability that a given mutation will actually manifest as disease. Some mutations have penetrance of nearly 100%, meaning practically all carriers develop the condition over the course of their lives. Others have low penetrance, making the prognosis far less predictable for affected families. This starts to get more interesting when we realize there's a fascinating gray zone between what we call disease and what we simply call difference. Color blindness is a classic example. It's technically a variation in color perception caused by changes in specific genes, but in many contexts it doesn't cause any significant functional limitation in the lives of those who have it. What defines a genetic variation as a disease rather than a difference? The answer usually depends on how much that characteristic interferes with the person's life within the environment they live in. Under certain historical conditions, some variants we now call diseases might have been neutral or even advantageous for the group's survival. Modern genetics constantly grapples with that tension. And there's a detail that changes everything here too. Human DNA isn't a static, unchanging sequence over the course of a lifetime. The genome undergoes thousands of small changes during a single person's existence. The vast majority of them are automatically corrected by cellular repair systems that operate with truly impressive efficiency. Only a tiny fraction of those changes persist and have any effect. When a mutation occurs in somatic cells, the ordinary cells of the body that aren't sperm or eggs, it can affect that specific tissue. But it won't be passed down to children. That's what happens in most cancers. Mutations accumulated over the years in cells that begin to divide in an uncontrolled way. The story is written within the organism, not in the generations that follow. When a mutation occurs in germline cells, those that will give rise to the gametes, it can be passed on to the next generations. This distinction between somatic and germline mutation is fundamental for understanding both individual risk and family risk of a genetic disease. It's the difference between a story that ends in one organism and a story that continues for generations. The amount of information contained in human DNA is hard to grasp intuitively. If the DNA from a single cell were stretched out in a straight line, it would measure about 6 feet. Multiply that by the roughly 37 trillion cells in the human body, and you'd reach a distance that would cover the round trip between the Earth and the Sun more than 100 times. All of that inside us, right now. All of that information is packed inside a cell nucleus about 6 micrometers in diameter, smaller than 1/10 the width of a human hair. This compaction is done in a highly organized way, using proteins called histones around which the DNA wraps itself. And that organization, in itself, influences which genes are accessible to be read and which remain silenced. What almost no one realizes is that only about 2% of the human genome corresponds to protein-coding genes. The rest, long referred to as junk DNA, contains regulatory sequences, repetitive elements, and regions whose function is still being investigated. Progressively, this material once considered useless has been revealing important roles that no one imagined before. There's a class of genetic diseases that doesn't involve any mutation in the classical sense. No swapped letter, no missing sequence. The problem, in these cases, lies in the number of copies of a gene or a chromosomal region. Some people are born with extra copies of certain sequences. Others, with fewer. Both situations can have significant consequences. These variations are called copy number variations, and they can cause conditions as diverse as autism, schizophrenia, cognitive deficits, or, in other cases, no apparent clinical manifestation at all. What determines the effect is which genes are in the affected regions and what the impact is of having them in double or reduced dose. The imbalance, not the sequence error, is the problem. This starts to get more interesting when we consider one of the most counterintuitive discoveries of modern genetics. A large part of the human genome is made up of sequences that came from viruses. Over millions of years of evolution, viruses called retroviruses infected germline cells of human ancestors and inserted their genetic material into the host's DNA. These sequences account for about 8% of the genome. Most of these ancient viral sequences are silenced and appear to have no active function today. But recent research shows that some of them were recruited by evolution for genuine functions, including the regulation of gene expression and even the development of the placenta in mammals. What seemed like an invasion became an essential part of who we are. A question that seems simple but has no direct answer: how many genetic diseases are there? The database that catalogues conditions with known genetic basis has already recorded more than 7,000 distinct conditions. And that number keeps growing as new associations are discovered every year, with sequencing technologies becoming increasingly accessible and powerful. It's estimated that about 80% of so-called rare diseases have an identified or suspected genetic origin. And rare diseases, despite the name, aren't all that rare in the aggregate. They affect approximately 300 million people worldwide. That means that collectively, rare genetic diseases represent a public health challenge of truly significant scale. And there's a detail that changes everything in this conversation: the word mutation almost always carries a negative connotation. Something that went wrong, something that needs to be fixed. But from a biological standpoint, mutations are simply changes. The vast majority of them are neutral, cause no perceptible effect, and go unnoticed across entire generations. Without mutations, there would be no genetic diversity. Without genetic diversity, there would be no evolution. The problem arises when a mutation hits a critical region of the genome, the gene responsible for an essential function, and significantly alters the protein that gene produces. In that case, depending on the role of that protein, the consequences can be sweeping or very localized. What determines whether a mutation will be harmless, problematic, or even advantageous is a combination of where it occurs, what it changes, and in which context it's expressed. There are different types of mutation, and each has a distinct mechanism and impact. The simplest type is the substitution of one nitrogenous base for another, one letter of the genetic code swapped for a different one. Depending on which letter was swapped and in which position, this can change nothing, slightly alter the resulting protein, or completely halt its production. There are also insertions and deletions, situations where one or more bases are added to or removed from the sequence. This type of mutation can cause what's called a frame shift, because the genetic code is read in groups of 3 bases. If a base is inserted or removed, all the following groups shift out of position, producing a completely different protein from that point on. There's also a less intuitive type, repeat expansion mutations. Certain regions of the genome contain sequences that repeat naturally. When those repetitions exceed a critical number, they can cause disease. And that's exactly the mechanism behind conditions like Huntington's disease and Fragile X syndrome, two very different conditions, but united by that same strange kind of error. What almost no one realizes is that each person is born with about 40 to 80 new mutations that weren't present in either parent. Mutations that arise during the formation of the gametes or in the earliest divisions of the embryo. For a genome with 3 billion base pairs, that represents an extraordinarily low error rate, the result of repair systems that work continuously inside the cells. When those systems fail, whether due to mutations in the repair genes themselves, the mutation rate increases dramatically. That's exactly the scenario in some cancer predisposition syndromes. Mutations in the BRCA1 and BRCA2 genes, for example, compromise a specific DNA repair mechanism, leaving the genome more vulnerable to the accumulation of errors over time. The system that protects the code begins to fail silently. Not all mutations arise spontaneously. External agents called mutagens can significantly increase the mutation rate in exposed cells. Ionizing radiation, certain chemicals, cigarette smoke, and even some viruses are known mutagens. What they have in common is the ability to damage DNA in ways that repair systems can't always fix with full fidelity. Ultraviolet radiation from the sun, for example, causes a specific type of damage, the fusion of two adjacent thymine bases. When that damage isn't repaired before the cell divides, the mutation is incorporated into the new copy of the DNA. The accumulation of this type of mutation over the years is one of the main mechanisms behind skin cancer. A story written by the sun, slowly, on every person's skin. There's also a category of mutation that challenges the idea of the genome as a linear, stable sequence. These are called transposable elements, jumping genes. They're DNA sequences that have the ability to move from one place to another within the genome itself. When they land inside a functional gene, they can completely disrupt it, silencing an instruction that was supposed to be active. About 45% of the human genome is made up of transposable elements of different types. Most of them are silenced and no longer move. But a small fraction remains active, and new insertions caused by these elements are responsible for a detectable proportion of sporadic genetic diseases, cases with no explainable family history. Hemophilia A has documented cases caused by exactly this mechanism. This shouldn't happen this way, but it does. There are regions of the genome that are intrinsically more prone to mutations than others. Called mutational hotspots, these regions accumulate changes at a far higher frequency than would be expected by chance. Part of this comes down to the local structure of the DNA. Certain sequences form secondary structures that make faithful replication more difficult. The TP53 gene, which regulates cell cycle control and is mutated in more than half of all human cancers, is located in a region with characteristics that make it prone to mutations. The gene that's supposed to be the guardian of the genome sits in one of the most vulnerable spots in the very genome it protects. There's a deep irony in that which biology doesn't resolve, it simply presents. There are mutations that don't affect the DNA sequence itself, but deeply compromise how it's read. These are splicing mutations. To understand them, it helps to remember that human genes aren't continuous stretches of instructions. They contain coding regions interspersed with non-coding ones. Before the instructions are used, the RNA that's produced needs to be processed, the unnecessary parts are removed and the useful ones are joined together. When a mutation alters the signals that guide this processing, the result may include parts that should have been removed, or exclude parts that should have been kept. The result is a protein with an altered structure, even though the main coding sequence was never touched. Splicing mutations are estimated to be responsible for about 10 to 15% of all known monogenic diseases. Some mutations are silent in the most literal sense. They don't change the protein produced. They don't cause any detectable symptom. This is possible because the genetic code is redundant. Several different codons can specify the same amino acid. Swapping a letter that doesn't change the resulting amino acid seems, at first glance, to have zero consequence. And for a long time, that's how it was understood. More recent research, however, has shown that even these apparently silent mutations can have subtle effects. The speed at which the ribosome reads the RNA, and therefore the speed at which the protein is assembled, can be influenced by the choice of codon. Different translation speeds affect how the protein folds. And misfolded proteins can have altered functions. What seemed silent on the surface can have consequences that only show up under specific conditions. There's a type of mutation that's particularly insidious because it can't be detected by the most common analytical methods, the mosaic mutation. It occurs when a cell undergoes a mutation after fertilization, during embryonic development. The result is an organism with two distinct cell populations, one carrying the mutation and one without it. Depending on when the mutation occurred, it may be present in many tissues or just a few. This creates clinical pictures that vary enormously in severity and are often not recognized as genetic at first glance. Mosaicism also complicates genetic counseling, because a parent who carries a condition in mosaic form can pass the mutation to children in its full form, meaning the children can be more severely affected than the parent who carries the condition. One of the most recent and surprising discoveries about mutations concerns aging itself. As cells divide over the course of a lifetime, somatic mutations inevitably accumulate. Tissues with high cell turnover, like blood and the intestinal lining, accumulate these mutations more quickly. And that accumulation isn't neutral. Recent research has shown that in people over 70, it's common to find clones of blood cells carrying mutations in genes associated with leukemia, without the person actually having leukemia. This phenomenon was called clonal hematopoiesis of indeterminate potential, and it appears to increase the risk of cardiovascular disease in addition to cancer risk. This suggests that aging is, in part, a process of accumulating somatic mutations. The boundary between aging and genetically getting sick is far thinner than once thought. The idea that traits pass from parents to children is ancient. But the explanation of how that happens is surprisingly recent. Gregor Mendel described the basic rules of inheritance in the 19th century, working with peas, without knowing DNA existed. His observations were ignored for decades and only rediscovered in the early 20th century, when cell biology began connecting the patterns he described with physical structures inside the cells. The central principle Mendel identified is that traits are determined by hereditary units that exist in pairs. Each person inherits one version of each gene from their father and one from their mother. The combination of these two versions determines which trait manifests. This applies to eye color just as much as it does to the risk of certain diseases. An elegant logic that reality, however, makes far more complex than it first appears. To understand how genetic diseases are inherited, it's essential to grasp the difference between dominant and recessive alleles. A dominant allele manifests even when present in only one copy. Inheriting it from just one parent is enough for the effect to appear. A recessive allele, on the other hand, only manifests when present in two copies, one inherited from each parent. This asymmetry has enormous consequences for how diseases spread through families. This means a person can carry a recessive disease-causing allele for their entire life without ever showing symptoms, as long as the other allele in the pair is functional. That person is called a carrier. The problem arises when two carriers have children together. There's a 25% chance that each child will inherit both recessive alleles and develop the disease. A probability that isn't always visible in the family history. And there's a detail that changes everything in dominant inheritance. Not all dominant diseases manifest the same way in everyone who carries the altered allele. Some dominant conditions have incomplete penetrance. The allele is present, but doesn't necessarily produce symptoms. Others have variable expressivity. Symptoms appear, but with very different severity among carriers within the same family. Neurofibromatosis type 1 is a clear example of this. All carriers of the mutation have the condition in genetic terms, but the clinical spectrum varies enormously. Some develop only characteristic skin spots. Others develop nerve tumors, neurological complications, and other serious manifestations. The difference between these presentations isn't just in the main gene, but in the full complexity of the rest of each person's genome and their environment over the years. Sex chromosomes introduce a different logic to the inheritance of certain diseases. Women have two X chromosomes. Men have one X and one Y. Genes located on the X chromosome are present in 2 copies in women and only 1 in men. This asymmetry creates a specific vulnerability for males that doesn't exist in the same way for females. If a man inherits a recessive disease-causing allele on his only X chromosome, he has no second functional copy to compensate. The disease manifests. A woman, on the other hand, can inherit that same defective allele on one of her 2 X chromosomes and still not develop the condition, because the other X chromosome can provide the functional version of the gene. That's the X-linked inheritance pattern, and it explains why conditions like hemophilia and color blindness affect far more men than women. There's a form of inheritance that defies the simple logic of dominant and recessive. It's autosomal dominant de novo inheritance. In this case, the mutation doesn't come from either parent. It arises spontaneously during the formation of the gametes or in the first divisions of the embryo. The result is a child with a dominant disease and no family history that could have alerted the parents. This mechanism is responsible for a significant fraction of cases of conditions like achondroplasia, the most common form of genetically based dwarfism, and Dravet syndrome, a severe form of childhood epilepsy. In both cases, the parents have completely normal genomes. The frequency of these de novo mutations is correlated with paternal age, because the cells that generate sperm continue dividing throughout a man's lifetime, accumulating errors over time. What almost no one realizes is that there's something even more unsettling: genomic imprinting. A mechanism by which certain genes are expressed differently depending on which parent passed them on. Under normal conditions, a person uses both copies of a gene. But in genes subject to imprinting, only one copy is active, and which one depends entirely on its parental origin. Father or mother. The outcome changes completely. Angelman syndrome and Prader-Willi syndrome are caused by the loss of the same region on chromosome 15. But the clinical picture is completely different depending on which parent lost that region. If the deletion came from the mother, the child develops Angelman syndrome, with severe intellectual disability and characteristic movements. If it came from the father, the child develops Prader-Willi, with an entirely distinct clinical profile. The same missing chromosome. 2 completely different destinies. Mitochondrial inheritance follows completely different rules from nuclear inheritance. The mitochondria, the organelles responsible for energy production in cells, have their own DNA, separate from the chromosomal DNA. And that mitochondrial DNA is inherited almost exclusively from the mother. During fertilization, the mitochondria that enter the egg alongside the sperm are actively destroyed. This means that all of a person's mitochondria came from their mother, who inherited them from her mother, and so on in an unbroken maternal line across generations. Diseases caused by mutations in mitochondrial DNA are always passed from mother to all children, regardless of sex. A father with a mitochondrial disease doesn't pass the condition to any of his children. It's a chain that runs exclusively through the maternal line. There's a pattern of inheritance that was only fully understood in recent decades: inheritance through repeat expansion. Certain regions of the genome contain short sequences that repeat. In healthy people, the number of repetitions stays within a range considered normal. When that number exceeds a critical threshold, disease manifests. And each generation can inherit even more repetitions than the one before. This creates a phenomenon called genetic anticipation, the tendency for these repetitions to expand even further over time. Children and grandchildren of carriers may inherit an even greater number of repetitions and develop the disease earlier and more severely than their parents. Huntington's disease follows this pattern. Fragile X syndrome and several spinocerebellar ataxias do too. In each of these cases, the family history shows a progression where the disease becomes more severe with each passing generation. Consanguinity, relationships between people with common ancestry, increases the risk of recessive diseases in a mathematically predictable way. The closer the degree of kinship, the greater the probability that both partners are carriers of the same recessive allele inherited from a common ancestor. In populations where marriages between cousins are culturally common, certain recessive diseases appear at much higher rates than in the general population. One of the most recent frontiers in the study of genetic inheritance is the understanding that certain patterns of risk can be transmitted without involving mutations in the DNA sequence. Epigenetic modifications, chemical marks that regulate which genes are activated or silenced, can, under some circumstances, be passed from parents to children. The experience of one generation can leave biological marks on the next. Studies in populations that went through periods of severe famine showed that the metabolic risk of descendants was altered even in generations that never directly experienced the famine. The grandchildren of people who suffered from hunger showed risk patterns for diabetes and cardiovascular disease that couldn't be explained by the inherited DNA sequence alone. The experience had left a mark that time hadn't completely erased. Monogenic diseases are those caused by changes in a single gene, and for a long time they were the central model for how genetic disease was understood. The logic seemed straightforward: one gene, one protein, one disease. Reality, as always, turned out to be more nuanced. But these conditions remain the most well understood genetically, and the first to benefit from advances in diagnosis and therapy. There are today more than 6,000 catalogued monogenic diseases. Individually, most are rare. Collectively, they affect about 1 in every 100 people, which makes them, in the aggregate, a health problem of considerable proportions. Some are benign and practically imperceptible in daily life. Others are devastating from the earliest years, completely shaping the existence of those who carry them and those who care for them. Cystic fibrosis is probably the most studied autosomal recessive disease in the Western world. It's caused by mutations in the CFTR gene, which instructs the production of a protein responsible for transporting chloride across cell membranes. When that protein doesn't function correctly, the body's secretions become far thicker and more viscous than normal. A microscopic change with consequences that spread throughout the entire body. The consequences affect mainly the lungs and the digestive system. In the lungs, the thick mucus creates an environment favorable to repeated bacterial infections that, over time, progressively destroy lung tissue. In the pancreas, the ducts become obstructed, compromising digestion. Without treatment, life expectancy was just a few years. With current treatments, many patients reach adulthood and beyond. The CFTR gene has more than 2,000 known pathogenic variants. This means cystic fibrosis isn't a single disease, but a spectrum of conditions caused by different errors in the same gene. This diversity of mutations requires different therapeutic approaches for different groups of patients. And each newly discovered variant represents a new challenge for clinicians and for affected families. Huntington's disease holds a particular place in the history of genetics, not only because of the severity of the condition, but because of how its genetic discovery transformed the field. It's an autosomal dominant disease caused by the expansion of a 3-letter repeat in the HTT gene. Above a certain number of repetitions, the disease inevitably manifests. And there's a threshold above which penetrance is 100%. What makes Huntington's disease especially troubling from a human standpoint is its timeline. The symptoms, involuntary movements, progressive cognitive decline, psychiatric changes, generally appear between the ages of 30 and 50, after many carriers have already had children and potentially passed the mutation on. A genetic test can reveal the diagnosis years or decades before any symptom appears. But not all at-risk carriers choose to take that test. This starts to get more interesting when you think about what it means to know or not to know. Choosing to take the test is a decision about how to live the time that exists before symptoms arrive. Choosing not to take it is equally a decision. There's no right answer, and genetics offers no simple comfort in that situation. It offers only information, and it's up to each person to decide what to do with it. Phenylketonuria is a rare example of a serious genetic disease that can be completely controlled by a simple intervention: diet. It's caused by mutations in the gene that produces the enzyme responsible for metabolizing phenylalanine, an amino acid found in nearly all protein-containing foods. Without the functional enzyme, phenylalanine accumulates in the blood and brain, where it's toxic to the developing nervous system. If the child is diagnosed early and placed on a diet rigorously restricted in phenylalanine from the first days of life, neurological development can be completely normal. Phenylketonuria was the first disease to be included in newborn screening programs, the heel prick test, precisely because the window for intervention is narrow and the benefit of early diagnosis is extraordinary. Today, in countries with universal screening, the severe form of the disease has virtually disappeared as a cause of intellectual disability. Sickle cell disease is the most prevalent monogenic disease in the world, affecting millions of people, with the highest concentration in populations of African, Mediterranean, and Middle Eastern origin. It's caused by a single amino acid substitution in the hemoglobin chain, a change in a single nucleotide that turns a glutamic acid into a valine. A microscopic change with consequences that travel through the entire body. That seemingly small swap has enormous consequences. The altered hemoglobin tends to polymerize when oxygen levels drop, deforming red blood cells into a sickle shape. These misshapen cells obstruct small blood vessels, causing intense pain crises, organ damage, and a range of complications over a lifetime. And the fascinating irony is that the disease-causing allele persisted in the population for a clear evolutionary reason. Carriers of a single copy have greater resistance to malaria. Duchenne muscular dystrophy is the most common genetic muscle disease in childhood, affecting predominantly boys due to its X-linked inheritance pattern. It's caused by mutations in the DMD gene, which encodes dystrophin, a structural protein essential for the integrity of muscle fibers during contraction. Without it, the muscle tears with every cycle of movement. The DMD gene is the largest known human gene, occupying about 2.5 megabases of the X chromosome. Ironically, that enormous size makes it particularly vulnerable to spontaneous mutations. About 1/3 of Duchenne cases arise without any family history, as the result of de novo mutations. The disease that most affects boys is also the one that most often appears without warning, without history, without any prior sign in the family. Marfan syndrome is a connective tissue disease caused by mutations in a gene that produces a structural protein found in tissues throughout the body. When that protein is defective, structures that depend on resilient connective tissue become compromised: the aorta, the lens of the eye, the long bones. The heart is one of the most critical targets, and progressive dilation of the aorta can lead to fatal rupture if not monitored. Marfan syndrome gained historical attention through the suspicion, never confirmed, that Abraham Lincoln may have been a carrier. Some musicians and high-level athletes have also been retrospectively identified as possible carriers, sparking discussions about how the body type associated with the condition may have conferred advantages in certain physical activities. The body that genetics shapes can be simultaneously vulnerable in one way and exceptional in another. Tay-Sachs disease is a fatal recessive condition caused by the absence of an enzyme responsible for breaking down a specific lipid in the lysosomes, the cellular structures in charge of discarding molecules. Without the enzyme, that lipid progressively accumulates in neurons, slowly destroying them over the first years of life. It's one of the most difficult diseases to follow clinically. Babies with Tay-Sachs appear normal at birth. Around 6 months, development begins to regress. Vision is lost, followed by motor and cognitive function. Most affected children don't survive beyond 4 or 5 years. Carrier screening programs within the highest risk community, launched in the 1970s, managed to dramatically reduce the incidence of the disease, becoming a model for similar programs in other populations around the world. While monogenic diseases involve changes in a single gene, chromosomal diseases operate on an entirely different scale. The problem isn't a wrong letter in the code. It's the structure or quantity of the chromosomes themselves. It's as if, instead of a typo in a sentence, entire pages of a book were duplicated, inverted, or simply missing. Human chromosomes each carry hundreds or thousands of genes. Any change in the number or organization of these chromosomes simultaneously affects an enormous number of genetic instructions. That's why chromosomal diseases tend to have broad, systemic effects, touching multiple organs and systems at the same time. The imbalance ripples throughout the developing body. It's estimated that about 50% of first trimester miscarriages are caused by chromosomal abnormalities, most of them incompatible with development beyond the first few weeks. The developing human organism is extraordinarily sensitive to chromosomal imbalances, and natural selection eliminates most of these changes before birth. What reaches the world alive is only a fraction of what is conceived. Down syndrome is the best-known chromosomal anomaly and the most common among live births, occurring in approximately 1 in every 700 births. In the vast majority of cases, it's caused by the presence of 3 copies of chromosome 21 instead of 2. The extra chromosome generally arises from an error in chromosome separation during egg formation. A silent error at the most intimate moment of biology. Chromosome 21 is the smallest human chromosome in terms of gene count. And yet, having 1 extra copy of those genes is enough to profoundly alter development. The correlation between advanced maternal age and the risk of trisomy 21 is one of the best documented in medicine. This occurs because eggs remain suspended in the process of division for decades, and the longer they stay in that state, the greater the likelihood of errors in chromosome separation. Beyond simple trisomy, there are lesser-known forms of Down syndrome that reveal important nuances about how chromosomes behave. In about 3% of cases, the trisomy isn't uniform across all cells. This pattern, called mosaicism, can result in milder clinical manifestations, depending on which tissues were affected by the trisomy and which preserved the normal chromosome count. Anomalies in the sex chromosomes form a diverse group of conditions with surprisingly varied clinical manifestations. Klinefelter syndrome, for example, affects men who are born with 2 X chromosomes and 1 Y, and is one of the most common chromosomal anomalies, occurring in approximately 1 in every 600 men. Despite its prevalence, it's remarkably underdiagnosed. It's estimated that only 25% of carriers receive the diagnosis over the course of their lives. Turner syndrome occurs when a person is born with only one functional X chromosome, without the second sex chromosome. The fact that humans can survive with only one X chromosome reveals something important about the biology of sex chromosomes. The inactivation of one of the X chromosomes in typical females means that, in essence, all female cells function with only one active X. Turner syndrome takes that principle to its extreme. There's a category of chromosomal anomaly that can be completely invisible clinically in the person who carries it, but has profound implications for the health of their children: balanced translocations. In this type of change, segments of two different chromosomes swap positions with each other, but no genetic material is lost or gained. The carrier has the right amount of genetic information, just reorganized in a different way. The problem arises during the formation of the gametes. When reorganized chromosomes attempt to separate correctly, there's a significant probability that the resulting gametes will receive an unbalanced amount of chromosomal material. This can result in repeated miscarriages, the birth of children with multiple anomalies, or unexplained infertility. Couples with a history of multiple spontaneous miscarriages frequently discover a balanced translocation as the explanation. The majority of diseases affecting the world's population don't follow the simple patterns of Mendelian inheritance. There's no single responsible gene, no clearly identifiable mutation, no 25 or 50% risk rule for children. These are called multifactorial diseases, conditions where genetics plays a role, but where environment, behavior, and time also have decisive and intertwined parts. Type 2 diabetes, hypertension, cardiovascular disease, schizophrenia, depression, autism, all fall into this category. Collectively, they're responsible for the majority of the disease burden in modern populations. And paradoxically, they're the least understood from a genetic standpoint, precisely because the underlying biology is far more complex than that of a disease caused by a single defective gene. The central concept behind multifactorial diseases is that of a susceptibility threshold. No single factor, neither genetic nor environmental, is sufficient to cause the disease on its own. It's the combination and accumulation of multiple factors that eventually crosses a threshold beyond which the condition manifests. This explains why siblings with the same genetics can have very different clinical outcomes when they live in different environments. One of the most powerful tools for studying genetic contributions to multifactorial diseases is twin studies. Identical twins share nearly 100% of their DNA. Fraternal twins share on average 50%, like any pair of siblings. Comparing the concordance rate of a disease between these two types of twins allows researchers to estimate how much of the variation in risk is explained by genetics and how much by environment. For schizophrenia, the concordance rate among identical twins is approximately 50%. That means that even with identical genomes, one twin can develop the condition while the other doesn't. Genetics is clearly important, but it's not deterministic. That 50% concordance is simultaneously strong evidence of genetic influence and an equally strong argument that genetics alone doesn't tell the whole story. What almost no one realizes is that the interaction between genes and environment is one of the most important and most difficult concepts to study in medicine. The effect of a genetic variant isn't fixed. It can be amplified, reduced, or even reversed depending on the environment the organism is in. The same gene, two completely different outcomes, determined by behavior and circumstance. The Pima, an indigenous group from the American Southwest, have become a classic case study. They show one of the highest rates of type 2 diabetes in the world when living on a Western diet. Pima groups living in Mexico, with traditional diets and lifestyles, have diabetes rates comparable to the general American population, despite the same genome. Genetics defines the field of possibilities. The environment determines where within that field each person ends up. Schizophrenia is one of the psychiatric conditions where the tension between genetics and environment is most evident and most studied. The heritability of schizophrenia is estimated at around 80%, one of the highest among psychiatric conditions. And yet, most people with schizophrenia don't have first-degree relatives with the condition. The genetics are there, but distributed in a way that isn't easily visible in family trees. The most widely accepted current model is that of vulnerability and stress. Genetics determines the baseline level of vulnerability, and environmental factors over the course of a lifetime may or may not push past the threshold for the disease to manifest. Two people with the same genetic risk profile can have completely different clinical trajectories depending on the circumstances they encounter throughout their lives. Inside nearly every human cell there's a structure that carries its own DNA, separate, compact, and with an evolutionary history completely different from the DNA in the nucleus. The mitochondria were, approximately 1.5 billion years ago, independent bacteria that were incorporated by larger cells in a relationship that became permanent and mutually dependent. That event left deep marks on the biology of every living cell. The human mitochondrial genome is remarkably compact. While nuclear DNA contains about 3 billion base pairs and approximately 20,000 genes, mitochondrial DNA has only 16,569 base pairs and encodes 37 genes. That compactness has a direct consequence. Any mutation in mitochondrial DNA has a very high probability of hitting a region with an important function. A unique characteristic of mitochondrial diseases is the phenomenon of heteroplasmy. Since each cell contains hundreds or thousands of copies of mitochondrial DNA, it's possible for some of those copies to carry a mutation while others don't. The ratio between mutated and normal mitochondria within a cell is called the heteroplasmy level. The higher that ratio in a given tissue, the greater the energy dysfunction in that specific tissue. This creates clinically puzzling situations. A person can carry a mitochondrial mutation with a low heteroplasmy level and show no manifestation at all, while a close relative with the same allele but a higher proportion of mutated mitochondria presents serious disease. The same DNA, completely different consequences. Here, quantity matters just as much as quality. Epigenetics profoundly challenged the view of DNA as a fixed, unchanging text. The term refers to modifications that affect gene expression without altering the DNA sequence itself. Like notes written in pencil over a printed text, changing the meaning without touching the original letters. Every cell type in the human body has essentially the same DNA, but completely different epigenetic patterns. What made epigenetics especially relevant to medicine was the discovery that these patterns aren't permanent or entirely individual. They can be influenced by the environment, can shift over the course of a lifetime, and in some cases can be passed between generations. Experience shapes the genome, and the shaped genome can be passed on. The most studied epigenetic mechanism is DNA methylation, the addition of a chemical group to a specific base in the DNA. When regions near genes are methylated, the cell's machinery has restricted access to that gene, reducing or silencing its expression. When those regions are demethylated, the gene becomes more accessible and its expression increases. A control system that works without changing a single letter of the code. One fascinating practical application of DNA methylation is the so-called epigenetic clock, a set of markers that allows estimating the biological age of a tissue with remarkable precision. People with methylation patterns indicating a biological age greater than their chronological age have, on average, a higher risk of age-related diseases. Biology ages at a different pace than the calendar. One of the most unsettling discoveries of modern epigenetics was the evidence that adverse experiences in childhood leave measurable molecular marks on the genome. Abuse, neglect, and chronic stress in the early years of life alter methylation patterns in regions of the genome associated with stress regulation. The trauma had left a permanent molecular signature. Experience transformed into biology. Cancer can be understood, in large part, as an epigenetic disease as well as a genetic one. Cancer cells display profound and systematic epigenetic changes. Genes that should be silenced are activated, while the brakes that should contain uncontrolled cell growth are epigenetically switched off. This epigenetic dimension opened up a new class of therapeutic targets that didn't exist before. This epigenetic dimension of cancer opened the possibility of reversing the silencing of tumor suppressor genes, reactivating the brakes that cancer had switched off. Drugs that modify the enzymes responsible for DNA methylation or for the modification of the proteins around which DNA wraps itself are already in clinical use. The epigenome, unlike the genome, is reversible. That, in itself, is a form of hope. Rare diseases form a paradoxical universe. Individually, each one affects very few people. Collectively, they affect hundreds of millions. The total number of catalogued rare diseases exceeds 7,000, and new conditions are described regularly. About 80% of rare diseases have an identified or suspected genetic origin. In the aggregate, they represent an invisible, and at the same time immense, public health challenge. The diagnostic journey for patients with rare genetic diseases is, in many cases, extraordinarily long. Studies across different countries estimate that the average time between the onset of symptoms and the correct diagnosis ranges from 4 to 8 years. During that period, patients often receive incorrect diagnoses, are subjected to inappropriate treatments, and accumulate damage that could have been avoided with earlier identification. Hutchinson-Gilford progeria syndrome is one of the rarest and most biologically fascinating genetic conditions. It affects approximately 1 in every 4 to 8 million births. It's caused by a specific mutation that results in the production of a truncated, abnormal version of a structural protein in the cell nucleus. The result is dramatic accelerated aging, with characteristics of old age appearing in the first decade of life. What makes progeria especially relevant beyond its own rarity is the discovery that the abnormal protein produced by the mutation is also produced in small amounts in normal cells during aging. This suggests that the mechanism causing the accelerated aging in progeria may be involved, on a smaller scale, in the normal aging of all of us. These children have become, paradoxically, a window into the biology of human aging. Williams syndrome is caused by the deletion of a small region of chromosome 7 containing about 25 genes. It produces a highly characteristic cognitive and behavioral profile that's fascinating from a neuroscience standpoint. People with Williams syndrome show deficits in visuospatial skills, but often have well-preserved verbal abilities, exceptional facial memory, and a remarkable sociability. This dissociated profile, deficits in some cognitive domains and preservation or even enhancement in others, made Williams syndrome a valuable model for understanding the modular architecture of the human brain. Each deleted gene in that region revealed something about how human cognition is genetically organized. And each revelation raises new questions about what it means to have a brain and what it means to be a person. Fibrodysplasia ossificans progressiva is considered by many to be the most disabling of known rare diseases. There are about 800 confirmed cases in the world. It's caused by a single mutation that causes hyperactivation of a signaling receptor involved in bone formation. Soft tissues like muscle, tendon, and ligament are progressively converted into bone. The body builds a second skeleton around the first. Any trauma, even a minor one, can trigger a new wave of ossification in the affected area. The attempt to surgically remove the extra bone triggers even more ossification. Patients frequently become completely immobilized before the age of 30. The molecular understanding of the disease, however, has opened promising therapeutic avenues. Inhibitors of the affected signaling pathway are in clinical trials and represent the first real hope of a disease-modifying treatment. Cancer is, fundamentally, a genetic disease, but of a particular kind. Unlike inherited genetic diseases, most cancers result from mutations that accumulate over a lifetime in specific somatic cells, without being passed on to children. It's a disease of the genome that happens inside a single organism, in a single tissue, over time. A silent evolution unfolding within the body itself. The central idea is that of somatic evolution, cells that accumulate mutations conferring a proliferative advantage tend to expand their clones, accumulating more mutations along the way. Cancer is Darwinian evolution happening inside the body itself, with devastating consequences for the host organism. The same mechanism that created all the diversity of life on Earth happens, in accelerated form, inside every tumor. The genes involved in cancer development fall into two functionally opposite categories. Proto-oncogenes are genes that, in their normal form, promote cell growth and division in a controlled way. When mutated in certain ways, they become oncogenes, pushing the cell toward constant division. Tumor suppressor genes function as brakes. For them to lose their protective function, typically both copies need to be inactivated. The TP53 gene, which encodes the protein often called the guardian of the genome, is the most frequently mutated tumor suppressor gene in human cancers, present in more than 50% of all cancer types. When it functions normally, P53 detects DNA damage and prevents the cell from dividing until the damage is repaired. Or it activates cell death if the damage is irreparable. When it fails, the genome's protection silently disappears. Mutations in the BRCA1 and BRCA2 genes are the best known in the context of hereditary cancer predisposition. Both genes participate in a DNA repair mechanism essential for correcting serious breaks in the double strand. When these genes are mutated, cells accumulate genomic errors at a much faster rate. Women who carry mutations in BRCA1 have a cumulative lifetime risk of breast cancer estimated at 55 to 72%. One of the most transformative discoveries in molecular oncology was the recognition that cancers of the same organ can be biologically very distinct from one another. Two breast carcinomas can have completely different mutational profiles, respond to different treatments, and have different prognoses. This led to a conceptual restructuring of cancer, from a disease defined by the organ of origin to a disease defined by its molecular profile. The approval of tumor-agnostic drugs, which treat any cancer with a specific genetic alteration regardless of the organ affected, marked a turning point in pharmaceutical regulation. Larotrectinib, approved in 2018, treats any solid tumor carrying fusions involving a specific gene, whether it's lung cancer, thyroid cancer, soft tissue cancer, or any other. The response rate in tumors with that specific alteration exceeds 75%. The disease redefined by its biology. Treatment resistance is one of the central challenges of modern oncology, and it has a fundamentally genetic explanation. When a drug eliminates most of the cells in a tumor, some cells with mutations that confer resistance to that drug often survive. With the selective pressure of treatment eliminating the sensitive cells, the resistant ones expand. Its Darwinian evolution accelerated inside the tumor, responding to the treatment that was supposed to destroy it. Tumor sequencing has revealed that some cancers carry characteristic mutational signatures, specific patterns of which bases were substituted and in what sequence context, that work like fingerprints of the processes that caused those mutations. Exposure to ultraviolet light leaves a recognizable signature. Tobacco smoking leaves another. Defects in DNA repair mechanisms leave yet another. Each exposure history writes its own signature on the tumor's genome. This catalogue of mutational signatures allows researchers to retrospectively reconstruct the mutational history of a tumor, which processes were active, at what point, and with what intensity. It's a kind of molecular archaeology of cancer. A way of reading, in the mutations present, the chapters of the story that led that cell to become malignant. Genomic editing with CRISPR-Cas9 has opened concrete prospects for treating genetically based cancers. In blood cancers, a patient's T cells can be genetically edited to express receptors that specifically recognize antigens present on tumor cells. In refractory leukemias that no longer respond to any conventional treatment, the rates of complete remission with this approach reach 70 to 90%. For some patients who had exhausted all other options, these represented lasting cures. One of the most compelling horizons in cancer genetics involves not just treating established tumors, but detecting them before they become clinically relevant. The concept of multimodal early detection, combining different types of molecular markers in a single blood sample, is advancing rapidly toward clinical application. The idea is to screen the entire organism in an integrated way, rather than searching for one cancer at a time. Recent studies have shown that it's possible to simultaneously detect signals from multiple types of cancer in a single blood sample, with specificity high enough to be clinically useful, and with the ability to locate the tissue of origin of the detected signal. This represents a profound conceptual shift in how we think about prevention and diagnosis. The arc of cancer genetics, from a descriptive science to a tool for early diagnosis and precision treatment, represents one of the deepest transformations in medicine this century. And this is where things become particularly fascinating. All of this science, all of this depth, all of this knowledge accumulated over decades, begins with the same thing. A cell. A nucleus. A text with 3 billion letters that silently carries the story of each person and of the entire human species. The code that makes us who we are, that unites us in our biology and separates us in our individual stories. Every advance in the study of genetic diseases is also an advance in understanding what it means to be human. Every gene discovered, every mechanism revealed, every treatment developed, is a partial answer to a question that no science answers completely. What we are, where we came from, and how far we can go. Genetics doesn't close those questions. It deepens them, making the mystery richer with every answer found. And as you let these thoughts slowly dissolve, as the weight of each piece of information is gently released, what remains is only the feeling that there's an immense world unfolding inside every cell, every second, in complete silence, of a world so complex it would take more than an entire lifetime to fully understand. And maybe that's exactly why it's so fascinating, because curiosity never reaches its end. It just grows softer, quieter, more like rest than like searching. And as you drift into a deep and restorative sleep, remember to subscribe, so these stories can keep finding you, night after night. Sleep well, and sweet dreams.